So good morning friends and my dear students, myself Professor Munde Sandeep, Head and Assistant Professor, Department of Microbiology, Sanjini Mahavidyalaya, Chapuli. So first of all, I welcome you in the YouTube class. In the last lecture, we already see the definitions, different definitions about the microbial staining techniques. Today we are going to studying the microbial staining techniques. There are different types of microbial staining techniques. These staining techniques, they are used for coloring the microorganism for observation under the microorganisms. This is first staining method, simple staining method. In the simple staining method, why it is called a simple staining method? Because it is very simple, because only one type of coloring agent or dye or stain they are used for coloring the whole microorganism or its parts. That's why it is called as simple staining methods. So in the simple staining methods, there are two types of simple staining methods: direct, or it is also called as positive staining methods. In these staining methods, there is staining of microorganism or object, and second one is the indirect, or we can also say that negative staining, in which background is stained. So we'll see details about the direct staining and indirect staining, what happens. So in the direct staining or positive staining method, what happens? In these staining methods, you have to take one slide. On the slide, you have to take any microbial sample with the help of an instrument called as neochrome wire loop, and on the center of slide you have to prepare the smear. Smear means whatever the sample you have to spread on the center of slide, and after spreading you just keep the slide into the air for air drying process, or sometimes you have to keep, or you have to just keep the slide above the gas burner for heat fixing process. Once the smear is dry, you have to add the stain. Only one type of any stains you have to add, for example crystal violet, for example B.Sc.F.Y or methylene blue, any one you have to add and keep for 2 minutes. After 2 minutes, you just remove the stain from the slide by inclining the slide in inverted positions, and after that, under the slow running tap water, you just keep the slide for removing the stains, and after that you have to air dry this smears. And after air drying, what happens? You have to add 1 drop of oil, cedar wood oil, and this slide is ready for observing under the microscope. You have to observe the slide under the microscope by using 100x, that we can say oil immersion objective microscopes. So this is the direct staining procedures. Under the microscope, what happens? We can observe microorganisms are colored. Microorganisms are colored and background remain colorless. In these staining methods, methylene blue, crystal violet, B.Sc.F.Y, these are the stains are used. It means that in the direct staining or positive staining, what happens? Only microorganism or its parts are stained, backgrounds remains colorless. In case of negative staining or indirect staining methods, what happens? This method is exactly opposite to the direct staining, because same procedure. You have to take slide, prepare smear. You just use another stains in place of positive stains. In this type of staining method, you have to use nigrosin or Indian ink stains on the smear. Same remove after 2 minutes these stains from the slide and air dry. Keep 1 drop of cedar wood oil on the smear. Observe under the microscope. Under the microscope we can observe what? Microorganisms are not colored, background is colored, microorganism remains colorless, they are just observing like the stars, and background is dark. These are the two different methods, direct and indirect, positive and negative, under the simple staining techniques. So friends, this is all about the procedure, techniques and observation of simple staining techniques. So now we'll see what is the principle of such staining techniques. This is very important. What is the main principle? Negative staining requires the use of acidic stains such as Indian ink and nigrosin. The acidic stains with its negatively charged chromosomes will not penetrate the cell because of the negative charges on the surface of the bacteria. So unstained cells are differentiable against the dark background since heat fixation is not required. The cells are not subjected to the staining effect of chemical and heat. Their natural size, shape and arrangements can be seen by this method. It is possible to observe bacteria that are difficult to stain. This is the main basic principle of simple staining techniques. So friends, this is all about the simple staining techniques. In the simple staining techniques there are two types, positive or direct, and second one is the indirect or negative staining techniques. So friends, this is all about these methods. In the next lecture we will see the another staining techniques. Till then, take care, stay at homes, wear the mask if you are outside the home, use sanitizers and keep the social distance, because we are belongs to microbiological students, and this is the microbiological virus infected to the peoples from one people to another, pandemic disease, and it's our duty to take the care and say or give the information to another peoples also. Till then, bye.